And right, we turn our attention now to cellular nutrition, and who better to give us the latest update than our next guest, who is a professor at the Department of Chemistry in Lomonov, Moscow State University. He's also the head of the Belozersky Institute for Physiochemical Biology at MSU. So please do join me in welcoming Professor Petr Sergiev, Moscow State University. A very warm welcome.
Hello, everybody. First of all, many thanks to organizers. It's a great pleasure to present an update on our project to this so bright audience. So today we had a lot of talk on global view on nutrition, on billions of tons of food. However, my talk will be at a very small scale. Then we should move to the scale of a single cell, because ultimately all the food we have is used to feed our cells. So cell have its own little stomach, which is called lysosome. In the lysosome, all the components, macromolecules, are decomposed to the monomers, which are used either as building blocks to synthesize other macromolecules, proteins, for example, or to molecules which may be used as a fuel, which is then utilized by energy plant of the cell, mitochondria, to produce energy, mainly in the form of ATP. So how nutrition can get to this cellular stomach, lysosome? One way is more or less intuitive from outside in a process which is called endocytosis. Another one is completely different. It is called autophagy, or self-eating. So the idea of autophagy is to decompose cellular components, such as macromolecular machines or even organelles. And there are two reasons why do we need autophagy. The one reason is because macromolecular machines, like all machines, like usual machines, can be damaged and broken. So in a cell, broken molecular machines are disassembled to provide fuel for the cell and provide building blocks for synthesis of new molecular machines. So this is one reason. Another reason is if we are starving, then autophagy may be used to destroy something which we don't really need in the cell, and to make things which we desperately need. What we also know is that autophagy efficiency is gradually declining with age. And mutations in the components of autophagy machinery may lead to several diseases, for example, Parkinson's disease. So there are reasons to activate autophagy. And why do we hope that activation of autophagy would help us? Well, actually, the reasons is that if we would constantly destroy molecular machine in the cell and then synthesize them anew, we would have, on average, newer molecular machines, so organelles, which would function better. Another reason is that for constant destruction and resynthesis of molecular machines, we would spend energy because we have excess of energy because we like to eat, yeah, sometimes excessively. This idea is not new, and there are many large companies, startups, and research labs who are working on the search for activators of autophagy. So today I will provide an update on our joint efforts between Lomonosov Moscow State University and EFKA company on the search for autophagy activators. How can we know that this or that compound really activate autophagy? We have several assays. The first one is dealing with a protein. Actually, I have to apologize for so many details, but I would try to make it more popular. So just one protein, which is named LC3. This protein is modified in a process of autophagy activation, and we can separate these protein isoforms. So if you have this lower band of LC3 protein, it means that autophagy is activated. Another assay is cell-based. So in this assay, we also utilize the same protein, which is linked to green fluorescent protein. So upon activation of autophagy, this protein gets to the lysosome and is degraded. So green fluorescence decreases, while the red one, which is used as a control, stays. So if we have activation of autophagy, cells became red but not green. So this approach is scalable to high throughput. So we then use it for high throughput screening. And then the next question, if we have assays, then where to search activators for autophagy? I already told you that the topic is not new and there are a lot of competitors. So why will we be any better than our competitors? So we have several ideas where to search for activators of autophagy. The first idea is pretty trivial. In chemical libraries, and we do high throughput screening of chemical libraries. Up to now, we screened up to 30,000 compounds. The next idea is to use bifunctional molecules, so-called AUTACs. So these chemical molecules, which are designed by us, they have, so to speak, two hands. The one hand touches autophagosome. Another hand would bring the substrate to autophagosome. So with these two hands, we can guide, for example, mitochondria for destruction in autophagosome. And here in the corner, you could see the example of such compounds. Actually, those two are not working, but we synthesized and screened about 50 variants and found several which are new and working. The next idea. In this conference, we already heard a lot about microbiome, because all of us contain several kilograms of microbes in our intestine. And those microbes actually help us because they are better chemists than we are. So microbes synthesize a lot of stuff, and actually microbiome and human being and animal being cooperate. So this is a kind of symbiosis. So we decided that probably microbiome from animals, which are naturally resistant to long starvation, might help those animals to activate autophagy, to survive starvation. So to this end, we obtained microbiome sample from those creatures. Actually, this is a photo of the actual camel, which provide us with microbiome. So we take microbiome from camels, from bats, which are hibernating in the cave in the middle of Russia, and also from bears, which rise after their hibernation. So we cultivated this microbiome, then test the extracts in those assays, which we already discussed. And since we see that activation is there, we may then purify compounds and identify them. And the final source of potential activators for autophagy is medicinal plants and different spices. So we took as many samples as possible, around 300 different plant species, which are used in traditional medicine, and do basically the same. So we extracted everything, tested, then purified the active compounds. And actually, we found quite a lot of activators of autophagy in the plants. Some of them are known. The known ones are shown here. But some of those are pretty new. So finally, we have several sources of activators for autophagy, and we collected, so to speak, short list of the best compounds for further testing. And what we can do next? Next, we can check the mechanism, because autophagy is really regulated in a complex way. Yeah, there are many branches of these regulatory pathways. I will not dwell into the details of this picture, but we know there are several mechanisms of activations. So we can use our molecular tools to understand what are the molecular targets for this or that activator. For example, this compound 19, whatever it means, label mitochondria for degradation, which we can see by immunoblotting. And we can see also activation of a protein sensor of starvation, AMPK. Then, actually in parallel with testing the mechanism, we also perform testing in animal models. So the first one is knockout mice, which we produced ourselves because we have mice facility in our institute and we can genome edit mice. So this model actually in a scientific sense it is not new. The gene is known, but we produced our own line for this project. This is mice with inactivated leptin gene. Leptin is a protein which is produced by our fat, and it signals us that maybe another cake would not be good for us. But those mice do not sense this, and they are constantly eating, so they became pretty huge. And another model we use is chemically induced Parkinson's disease model on zebrafish. So with this model, we tested this short list of compounds, and some of those are really working. You can see this LC3 assay, whatever it means. The lower band is thicker than upper one upon induction. So we can see activation of autophagy in zebrafish. We can see activation of autophagy in mice, for example, with the same AMPK, which is a sensor for starvation. Mice, at least they do not die, they feel good, and we measured their performance, such as endurance and strength. So they are pretty much okay. As for endurance, they are even fitter than control. And we compare with known activator of autophagy urolithin, which is U here. So everything seems fine. And to sum up, we have several sources where we search for activators of autophagy. We have several assays to measure autophagy. Then, if needed, we separate active compounds, identify them with many means, and test their mechanism of action, and test those activators in animal models. So that's all for today, and thank you for your attention.